Buenas tardes, ¿me escuchan? ¿Me escuchan? ¿Sí? Buenas tardes, vamos a dar inicio a nuestro seminario. Realmente es un placer darles la bienvenida al primer seminario de la serie de Frontiers de nuestro segundo semestre de este año. Y pues tenemos una invitada de primer nivel, la doctora Margarida Cardoso, la doctora es bióloga de la Universidad de Porto, recibió su grado en 2003 con honores. Después realizó un doctorado también en biología evolutiva, también en la Universidad de Porto, en el 2009. Y a partir de ahí inició su carrera científica con 3 postdoctorados consecutivos. Uno en Cornell University en Estados Unidos, del 2009 al 2012. Después se movió a la Universidad de Lausanne con una beca de la Fundación Marie Curie, del 2003 al 2015. Del 2016 al 2020 hizo su tercer y último postdoctorado en la Universidad de Heidelberg, en Alemania. Y a partir del 2021 ya tiene su propio grupo de investigación en el Instituto Francis Crick en United Kingdom, en Inglaterra. En su corta pero productiva carrera, Margarida ha recibido diferentes premios, reconocimientos a su trabajo. Evidentemente, todas las estancias postdoctorales fueron financiadas por diferentes fundaciones como la Novartis Postdoctoral Fellowship, una fellowship postdoctoral por la Fundación GABA en Portugal. También ha recibido, es miembro de la Academia Científica de Heidelberg. Tiene una participación activa en educación, además de dar cursos básicos. Ha formado recursos humanos, alumnos de licenciatura, doctorado, maestría y ya también postdoctorados. Participa en diferentes comités académicos y solamente voy a mencionar que es revisora de los grants de la Comunidad Económica Europea, de Inglaterra y de Francia. Y que es revisora de revistas de alto impacto como Nature, Cell, Nature Genetics, etcétera. Y bueno, todo el enfoque que siempre ha tenido Margarida en su estudio es entender evolución y desarrollo. Y prácticamente el foco de su grupo actual y que acarrea como consecuencia de su carrera es entender cómo puede relacionar el desarrollo embrionario y la evolución. Y en ese tema, pues ha publicado más de 20 artículos, otra vez en revistas muy buenas como Nature y Science. Y realmente le agradezco a Margarida que haya aceptado la invitación desde el primer correo. Y hoy nos va a presentar su seminario ya un poco más con las líneas que está desarrollando ya en su propio grupo, que obviamente tienen que ver todavía con evolución y desarrollo. Thank you very much for being here and for your seminar.
Thank you so much. Yes, I have to start by thanking you, Gustavo, in front of everyone for the invitation. It's such an honor to come to UNAM. You guys know that you have one of the most famous programs in genomics. Your students are known worldwide. And so when I got Gustavo's email, I couldn't be more thrilled about the invitation. So thank you so much. And I also want to thank the students for the wonderful time they showed me yesterday. And indeed, all the fame that is due to UNAM students is really justified by your enthusiasm, by your excitement about learning science, for everything you do. So thank you so much. So I'm going to tell you a little bit today about the work in our group studying organ evolution. So how organs originate and then how they diversify in form and function across species. And I'm going to do that by telling you 3 stories. So I'm going to start by telling you our work about an aspect of organ evolution that has been long overlooked, and that is the evolution of sex differences. And this is work that we wrapped up at the end of the year, so this will be a complete story. But then I want to share with you 2 other stories that are still ongoing, that we're actively working in our group, that try to ask how do new biological structures arise during evolution. So we try to understand how are new types of cells created, and then how are whole complex organs, how do they appear, and then how do they emerge in evolution. So then let me start with the first topic. And I want to highlight here Leticia Rodriguez Montes. She was the master's student, now PhD student, who did most of the work that you'll be hearing today. So sex differences are widespread across animals, and mammals are no exception. Many of the sex differences are visible to the naked eye. We'll have some examples here in the slide, but many of the differences are not. And that's because those differences exist in our internal organs. And even though those differences are not visible, they are no less important. Now, I'm not going to lecture you on the importance of studying sex differences. I think maybe you know this better than I do. I'm going to try to explain to you why an evolutionary biologist is so interested in this question. So what has long fascinated me about sex differences is how quickly they evolve across species. So people have studied sex differences in organisms as different as flies and butterflies and stick insects and frogs. And irrespective of the group of species they were examining, they realized that the genes that differ in expression between males and females evolve very quickly across species. And by this, I mean that when you look at two species, even though they are closely related, the genes that differ between the sexes are not the same. And it has been, for me, something, a little bit of a paradox, how these genes evolve so quickly. And that led me to want to study them in mammals. So this is the problem we're trying to solve. We're trying to understand differences, say, between what makes a male mouse brain different from a female mouse brain, but comparing it also with differences in human brain. And so this is a problem of linking genes all the way to phenotypes, in this case, the sex differences. So one way to approach it is to use gene expression, so the transcriptome as an intermediate step. And you can see already the transcriptome as a phenotype, albeit a molecular one. And the framework that you have here represented in the slide is one that has been used in the past. So it's using this framework that people realized in different organisms that differences between the sexes evolve very quickly. So what is it that we're bringing you to this research? Well, as Gustavo alluded, we bring in development. And that is because even if the phenotype that you're interested in understanding is a phenotype that manifests mostly in adults, and it could be argued that sex differences, is a good example of that, we know that these differences are established at some point during development. So understanding how these differences emerge and then how they differ between species requires to look at all the way through the development of the organ that we're interested in understanding. To understand sex differences, though, sometimes we need to move levels from organs all the way to cells. And that is because, say, we find an organ that has high levels of differences between the sexes. We call this sexually dimorphic. What does that mean? Does it mean that all cells in that organ differ between the sexes? Or are those differences specific to a particular cell type? In order to answer this question, we need to move between organs and cells. And the framework that you have here is the one that we decided to apply to our study. So we decided to combine evolution and development to try to understand how sex differences evolve so quickly. So what did we decide then to study in particular? First, we wanted to know how much sex differences are there when we look at different organs across different species. Then, when do these differences first appear during organ development? When is their onset? And then combine these two levels of information to start studying the evolution of sex bias expression. Then we're going to address the question that I already touched a little bit ago, which is, so when we find an organ that is sexually dimorphic, does it mean that all cells are sexually dimorphic, or does it mean that it's just a few cells are? And if so, are these the same cell types across species, or are these different ones? And finally, we're trying to understand the molecular basis of developmental sex differences. And the hope is by understanding what underlies these differences, we can then go back and understand why they're evolving so quickly. So this is what we set ourselves to do. And to study the evolution, we decided to focus on these species. So we have human, rat and mouse, rabbits, a marsupial, and we have here a chicken to help us identify the features that are mainly specific to mammals from characteristics that might be shared across all vertebrates. Now, this lecture is going to focus on mammals, but birds are really a very special system to study sex differences because they rely a lot less on hormones for their sex differences than mammals do. But that would be something for a very different lecture. So this lecture is going to focus on mammals. And we're going to look at evolutionary sex differences in a few somatic organs. So 2 regions of the brain, the cerebellum and the rest of the brain, which I'm just going to call brain, the heart, the kidney, and the liver. And then the strength of this project is that we're going to look not just at adults, but we're going to look at sex differences all throughout development. So we have time series that we create for each species in both males and females. And so what you have here is a time series for mouse. We created a similar time series for the other species. The only exception is humans, right? In humans, we had fewer replicates, and our data stopped a little bit after birth. For humans, though, we have a lot of data already for adults as part of the GTEx data set. So we're going to combine these 2 different types of data. But because the data for humans and the data for the other species was not collected exactly the same way, I'm going to start by showing you data for the other mammals, and then, but in a couple of slides, I promise, I'll come back to humans. So what did we find in terms of how many differences are there when we look at different organs in different species? So what I'm going to show you here is on the x-axis, the number of genes that differ in expression between males and females, which we call sex biased. You have the data for the different species on the y-axis, and then the different colors correspond to the different organs. So when we looked at mouse, we found that 3 organs, the 2 brain regions and the heart, have very low levels of sexual dimorphism, but then the kidney and the liver had a very large number of genes that differ between the sexes. Okay, people had looked at sex differences in mice before, and this is exactly what they found. So this was reassuring. But then what did we find when we look at the other species? Well, when we look at rats, we had a similar pattern in terms of the distribution of the organs, but overall, much fewer sex differences. So again, the kidney and the liver are the sexually dimorphic organs, but overall, we have a smaller number of sex biased genes. And then what happened when we got to the rabbits? Well, something completely different, right? Much lower levels of sexual dimorphism in the organs that we chose to study. But now we no longer see the kidney and liver with high numbers. If anything, the heart is the most sexually dimorphic organ. We went to the marsupial opossum, and again, we found very different patterns from what we had found in the rodents. And something else also when we look at chicken. Now, chicken, I have to explain you why the data are split in this way. So while in mammals, we have dosage compensation, which means that even though females have 2 X's and males only have 1 X, only a few genes on the X chromosome actually differ in expression between the sexes. The same is not true for birds. And so the genes that are located on the sex chromosomes of birds, most of them are not dosage compensated. And these are the ones that I mark here by adding these black dots. So hundreds of genes in every cell in a chicken body are sexually dimorphic. But in addition, because they are located on the sex chromosomes, but in addition to them, you can see there are hundreds of genes located in other parts of the genome that also show a lot of sex differences. So indeed, birds and mammals have very different patterns. Okay, so that's how many differences we found between the sexes. But then when did these differences start during development? So when we started looking at the profiles, and again, keeping in mind that we always have this time series, we realized that we could quickly summarize our data set into 3 main patterns. We could classify genes as being always sex biased, and that meant that irrespective of the time when we looked, they were always more expressed in one sex than the other. So they had these parallel trajectories, and we call them always sex biased. Then we had a second set of genes that differ before sexual maturity at some point. And then we had a third class of genes that had similar expression between the sexes all throughout development up until we reach sexual maturity. And then at that point, the differences between the sexes start. And when we look at all these genes and their prevalence in the different species, basically the whole majority of genes are in this class, okay? So most sex differences really only emerge with the start of sexual maturity. So let me show you the data in more detail for the mouse, just as an example. So what you have here for the different organs are, again, the number of sex-biased genes, very few in the 2 brain regions in the heart, a lot more in the kidney and the liver. And the dark colors correspond to the always sex-biased genes. So the genes that are sex-biased in the brain and cerebellum are sex-biased throughout the entire development of these organs. But when we go here to the 2 very sexually dimorphic organs, we see that most genes come from this category, okay? So they are similarly expressed between the sexes up until sexual maturity, when we have the onset of sex hormones, and then they differ between the sexes. So this was actually one of the most surprising results, or at least to me, it was one of the most surprising results of this project. So what I'm showing you here now is on the x-axis, the number of genes that are sex biased, but now only prior to sexual maturity, okay? So I'm excluding the genes that are sex biased, starting only after sexual maturity. And so when in the previous chart, the x-axis went from 0 all the way to 2000, now this x-axis goes from 0 to 50. This means that across all the species in our data set, and now I've added the data for humans too, and again, the different colors correspond to different organs, only a very small number of genes start being differentially expressed between the sexes prior to the onset of sexual maturity. So this is surprising to me because my expectation was that indeed, as development progressed, I was hoping to see more and more, or was expecting to see more and more sex differences. But what we really found is very small levels and then this abrupt start with the onset of sexual maturity. Now, despite this being a small number of genes, they involve a lot of transcription factors, and we do think they are very important in setting up the differences that happen later on. Now, among these few genes are these genes that are always sex biased, okay? So irrespective of the time when we look, they always differ between the sexes. And this set of always sex biased genes is very special. So most of them are genes that are located on the X chromosome, but they still have their counterparts on the Y. And these are genes that escape X chromosome inactivation. And these are the genes that are basically shared across mammals. But other than that, we didn't see a lot of conservation. So let's tackle that directly, right? Because that was the whole goal of this project, is to understand the evolution of sex bias expression. So let's go back to that initial plot that I showed you. Already when we had this first result, it was obvious there was very little conservation because the numbers are so different across the different species. But we decided to try to understand this better, and we focused in the mouse and rat because these are the most closely related species in our data set, and because these are the ones that have a similar profile, with more sex biased genes in the kidney and liver and very few in the other species. Sorry, this was what I should have said before, that each species in a way has its own set of sex biased genes. So then now let's look for mouse and rat. We decided to look at this more carefully, and so we decided to do this comparison. So we say, okay, let's take the genes that are sex biased in the mouse kidney and in the mouse liver and ask, are they also sex biased in the rat? And for less than 20% of the genes that are sex biased in mouse, they're also sex biased in the rat. And so what happens to the other genes? Well, in the kidney, most of the other genes are also expressed in the kidney in the rats, but they're expressed at just equal levels. And then we had a small proportion of genes that we couldn't really compare because they didn't have a one-to-one orthology between two species. So we didn't know, they didn't have a counterpart on the rats. Now, the reason that I'm showing you the data like this, separating genes that are one-to-one orthologs from those that are not, is that when we went to the liver, we found something a little bit different. So we found that we had a much larger number of genes that were sex biased in the mouse, but had no counterparts in the rat genome. So we wanted to understand this a little bit further. And so we decided to look at sex biased genes in terms of when did they first appear during evolution. So what you have here on the y-axis is exactly that, the age of the genes. And at the bottom, you have genes that were already present in the common ancestor of vertebrates. And then as you go up on the y-axis, the genes become progressively younger. And so at the top, you have genes that are private to mice. Okay, so they are not shared with rats or other kinds of murids. So the distribution of sex biased genes in the kidney is very similar to the distribution of genes that we see genome-wide for any kind of gene. But for the liver here, I hope you can see here is that we have an excess of genes that are sex biased in the liver, and they're very young. And we knew that we were severely underestimating this number because for genes that were sex biased in the liver, in the mouse, for 10% of them, we couldn't even assign an evolutionary age. That's because the evolutionary history was so complicated that we didn't know exactly when they had originated. So we said, okay, so what are these very, very young genes doing and why are they sex biased? So very young genes, evolutionarily speaking, are genes that are very poorly studied. So in order to understand what these genes were doing, we had to look at them one by one. And when we did by this, we realized that actually most of them came from expansions of only 3 protein families: the cytochrome P450, a transporter family, and then a major urinary protein family. Okay, So what do these families do, and what is their relationship with sex bias expression? Now, the cytochrome P450 family is a very large family. They have, the proteins, the enzymes do a lot of functions. But for our purpose here, it's important to note that they're involved in the detoxification and activation of xenobiotics, but they also are involved in transformation of endobiotics, like steroid hormones, which are very important for sex differences. Then the transporter family that you have there is involved in the transport of sex hormones. But my favorite family is by far that of the major urinary protein families, because this is what they do. They basically code for proteins that are put in the urine of mice, and same happens in other rodents, and they code for male-specific pheromones and other compounds that allow mice to communicate with each other, for males to compete with each other, for recognition of members of the same species and also members of different species. And yes, and this was very important characteristics, because when we see these differences between the sexes, there's a lot of people wonder how functional these differences are and how meaningful they are. But when we see characteristics like that, these types of functions enriched in our data set, we think this means that these differences in gene expression are very meaningful, and they're probably being driven by positive selection. Then we did similar work as I described here, but now from the perspective of the rat. And we again found an excess of genes that are private to the rat, so not shared with mouse. And when we went and looked for those families, we realized that they are part of the same families. So these families independently expanded after mouse and rat split, independently acquired expression in the liver and very high levels of sexual dimorphism. And again, we have an example of parallel evolution, which again further strengthens the idea that the sex differences that we are observing are driven by natural selection. All right, so now I'm going to shift gears a little bit. We had figured out that in the mouse and the rat, the sexually dimorphic organs are the liver and the kidney. But now the question was, is this something that is shared by all cell types in these organs, or are these differences specific for particular cell types? And so in order to answer this question, we had to move from looking at the tissue as a whole, the organ as a whole, and look at the different cells that make up these organs. And so what you have here is a single cell data set for the mouse liver. And the different colors respond to different cell populations. For the purpose of this talk, we were asking, okay, where are genes that differentially expressed between the sexes expressed? So we are going to call genes that are more highly expressed in males, male biased genes, and genes that are more highly expressed in females, female biased genes. And in both cases, we saw that they were both expressed in this group of cells, which are basically the hepatocytes, which is the main cell type of the liver. Then in this data set, we collected data for both males and females. So we have male hepatocytes and female hepatocytes. And we asked, what is the pattern for these sex biased genes? And what we observed is exactly what you would expect from the signal we see in the bulk, is that when we look at male cells and female cells separately, male biased genes are genes that are more highly expressed in the different types of male hepatocytes compared to females. And the female biased genes are those genes that are more highly expressed in female hepatocytes compared to male hepatocytes. Great. So we learned that sex bias expression in the mouse liver is mostly derived from the hepatocytes. Then we ask, okay, but what is happening in rats? Because most of the genes that are sex biased in rats are not the same that are sex biased in the mouse. Are we going to find the same cell types or a different one? In order to answer this question, we analyzed an existing single cell data set that had both male and female cells for the rat liver. Now, this is a special data set because even though hepatocytes are the most common cell type in the liver, they were trying to enrich for any other type. But they still had hepatocytes here. This is the the yellow group that you see over here. And then when we ask where are our sex bias genes expressed, again, in the hepatocytes. And as we had seen in the mouse, the difference is really because male bias genes are more highly expressed in male hepatocytes, and then female bias genes are more highly expressed in female hepatocytes. Okay, different genes between mouse and rat, same cell types. Now, what is happening prenatally? Now, prenatally, I told you there are no sex differences. True. But my question is, are these genes then not expressed at all? Are they only expressed later in development when we see the sex differences? Or are they already expressed? And if so, where are these genes expressed? So we analyzed a developmental data set for the mouse liver, again, male and female cells. Now, the liver is very different before birth because it's a hematopoietic tissue, so we have a very different cell composition. But then we asked, where do we see expression of our sex bias genes, those that are going to differ between the sexes in adults? And we have them here, and this is the group, is the hepatocytes. So what does this mean? This means that even before sex differences are established, these genes are already specifically expressed in hepatocytes. But at this point we wondered, okay, so maybe there are actually differences between the sexes, but maybe we're not able to tell them using our both data sets. But that's not right. So as I said, we had data for both males and females, and despite the fact that these genes are specifically expressed in hepatocytes, prior to birth, they express at similar levels between the sexes. So these genes are already specific to hepatocytes throughout most of liver development, but then it is only with the onset of sexual maturity that they start differing in expression between the sexes. So this is what we found for the liver then, that in the liver, the hepatocytes are the cell type that is sexually dimorphic. So there's a male version of hepatocytes and a female version of hepatocytes. What is then going on for the kidney? So we did exactly the same work that I just described for you for the liver, but instead of going through 3 new slides, I'm just going to give you the summary. And the summary is that in the kidney, which this is the structure you have here, there's also one cell type that is sexually dimorphic, and this is called the proximal tubule cells. They are located in this part of the kidney, and there's a male version and a female version for them. We look in mouse and we look in rats, and we find exactly the same cell types. So just like we had seen in the liver, different genes, but the same cell types. And importantly, for the kidney, we also know that the proximal tubule cells are also the sexually dimorphic cell type in humans because of existing data. So what I want you to take, one of the messages that I would like you to take home is that, yes, when we think about the evolution of sex differences, they evolve very quickly. We are talking mostly about different genes in different species. However, it is likely that the same cell types tend to be sexually dimorphic. And there's only one part then to wrap this whole thing up. We wanted to understand then what is the molecular basis, what leads to these differences in the expression between the sexes. And for the liver, we are very fortunate because people had pretty much figured out what are the main drivers of sex differences in the liver. And it actually starts with the growth hormone. And the growth hormone is produced continuously in female livers, but in male livers, it is basically pulsatile, okay? So this is difference in the pattern of the growth hormone that then activates the STAT5 dependent cascade in a different way. So when you have pulsatile hormone, the cascade is activated in an intermittent manner, whereas in females, it's activated in a persistent manner. And this difference in the timing then activates different transcription factors that then lead to sex bias expression. So then what we decided to do is to focus on data on all of these transcription factors. And we wanted to ask if with these transcription factors alone, we could explain most of the sex differences that we were finding. We also decided to look to 2 additional transcription factors that are responsive to sex hormones that had been also implicated in sex differences. So how do we do this? Well, we analyze a very large number of ChIP-seq data sets for both males and females that tell us where each of these transcription factors bind in the genome. And then we ask, how are they associated with our genes that are male biased and female biased? So I'm just going to show you the summary for this whole data set. So what you have here on the x-axis is data for the different transcription factors. And then we have our set of female biased genes and male biased genes. And every time you see a green dot, it means that indeed these transcription factors are significantly associated with the genes that we found. And so what did we find? We find that all the transcription factors that are under the control of the growth hormone are significantly associated with our sex biased genes. And in males, the same is true for transcription factors that are under the control of sex hormones, but not in females. Now, one of the transcription factors that you have here is STAT5B. Now, this result was not so meaningful because this is the major transcription factor in the liver. So pretty much STAT5 associates with all genes. So what we decided to do is to look at STAT5 in a more specific manner. But before I do that, I just wanted to also show you that as you'd expect, we look at the distribution of all of these transcription factors. We ask which cell type they are expressed, and they're all specifically expressed in hepatocytes, okay, which is the sexually dimorphic cell type. The exception then is the STAT5 because STAT5 controls pretty much all of liver development. So in STAT5, we actually had to examine a data set that told us what are the binding sites for STAT5 that are specifically open in females and in males. And as you'd expect, the STAT5 binding sites that are specifically open in females are the ones that are significantly associated with our female bias genes, and then the opposite for the male bias genes. So here's an important point, is that this is not just about transcription. It all has to do also a lot about chromatin accessibility and the regions of the DNA that are open in each of the sexes. So we then decided to look at this a little bit more closely by looking at a bunch of chromatin marks that tell us what are the regions that are closed and open in the different in males and females. And what we found here, once again, is for most of these marks, when we had open chromatin associated with female, so open chromatin specifically in females, it was associated with female bias genes. But for example, we had also marks where were specifically closed in males, and this was also specifically associated with females. So all in all, we learned that about 80% of the differences that we found in liver between the sexes could be explained by these transcription factors and chromatin features. But to me, the most important lesson was how highly combinatorial this was. So we never found sex bias gene just basically associated with one of these transcription factors. They were usually associated with multiple transcription factors. And when we compare genes that are sex biased only in mouse or in rats, what we figured out is just if you lost one of these transcription factors was enough to stop that gene from being sex biased. So it's relatively easy from a genetic point of view to create binding sites that respond to the sex hormones, which means that it's relatively easy to create variation on which natural selection can act. We did similar work like this to the kidney. There much less is known about sex differences. We had identified transcription factors in our data set that were differentially expressed between the sexes, and using those transcription factors, we're able to explain about 60% of the differences that we found between the sexes. In conclusion, this is what we learned from doing this work, that the extent of sex differences differ considerably between organs and species. So even when I told you that we found very low levels of sexual dimorphism in the rabbit, you have to take that with a grain of salt, because that's only true for the organs we decided to study. So we decided to look at the lungs or decided to look at adipose tissue, we might find something very different. And this is something that people need to have into consideration when studying sex differences. That the onset is basically associated with sexual maturity. So it's a little bit of a simple phenotype where with the onset of hormones during sexual maturity, that most differences in expression then lead to differences between the sexes. And that the sex differences in these organs are actually limited to specific cell populations. And these cell populations tend to be conserved across species. So for evolution, is what I told you before. There's a very few sex bias genes that are conserved across species, but we do have the same cell types being sexually dimorphic, which I think this is very good because it does mean that we can study sex differences in humans by using other animal organs, even if we have to be more careful when studying sex differences than when studying other phenotypes. So what is the driving force of these sex differences? I think there's a lot of evidence here that is really natural in sexual selection, so that these sex differences are really meaningful and are important in how these species adapt to the environments where they are. And in terms of the molecular basis, it's relatively simple in what comes to explaining a phenotype. We have transcription factors that respond to hormones, then are differently expressed between the sexes. That together with chromatin accessibility explains most of the sex differences we find. And that is very easy, as I said, to gain and lose binding sites. So it's very easy to both create sex bias expression and to lose sex bias expression. And we think it is because it is so simple genetically that we see it evolving so quickly across species. Okay. So that is for the complete story. And now I'm going to shift gears. And now you're going to hear about two stories that are pretty much in the midst of it. But I'm very excited about them. And while we are for sure not done, I think we've already made important advances. So my lab is working on what has to be one of the oldest problems in evolutionary biology. How do new and complex structures originate? Think about cells, think about tissues, think about organs. And why do I say this is an old question in evolutionary biology? Because this question was posed by Darwin himself. And Darwin realized that this would be one of the hardest things we would have to explain as evolutionary biologists. And Darwin gave the eye as an example of something that's very complex, made of different parts, but it only works when these parts are assembled together. I mean, we know that evolution is very much stepwise. So how do we explain with little steps, the emergence of new structures. In my lab, we address this question by asking, how do you create a new type of cell? But we don't shy away from the real question, because the hard question is not really how do you create a new type of cell. The real question is, how do new organs evolve? Because an organ is made of different cell types, they're organized in different tissues, which itself are organized to make this organ. So that's the definition of a complex biological structure. So if this is, okay, I hope you all agree that this is an important question, but I just told you that this is a question that has been plaguing evolutionary biologists for a long time. So why do I think now we are in a good place to answer this question? I think it's because what we were lacking were the tools. Okay, technology matters a lot. And what we now have are single cell and genomic technologies, which basically allow us to study individual cells in any organ of our choice, and importantly, in any species of our choice. And why does this matter? Because you could say, but we've been able to do this using other technology for a very long time. Well, this matters because with this data, we can have molecular data. And with molecular data, we can actually reconstruct the evolutionary history of cell types and organs. And this is really important because we already actually have a lot of theoretical models, ideas about how new cell types might emerge. We have much less theoretical understanding of how organs would come about, but we already have the ideas. We now have the tools. So what are we missing? In my opinion, the only thing we're missing, although it's the most important thing, it's empirical models. We need to find instances of novel cell types and novel organs that we can study. And here lies the big challenge. Okay, so some people obviously are interested in asking, say, how the vertebrate brain emerged, like how the neurons first appeared. And if you're interested in explaining how neurons first appeared or cardiomyocytes, then these are events that happened hundreds of millions of years ago. We have to go all the way back to the Cambrian explosion. And this is a very hard problem to deal with because evolution never stops. Okay, so from the moment when a new biological structure emerges, it keeps being transformed and changed through evolution. So if we hope to reconstruct the steps, the genetic, developmental steps, and evolutionary steps that led to the emergence of that structure, we need to find something that emerged relatively recently. So in my group, we are not so much interested in how a specific cell type came to be, but how cells in general emerge. And for that, we really were trying to find examples of cells and organs that evolved only recently in evolutionary time. And that was the challenge for us. And so this is where we decided, we thought it was a good place to search. So in mammals, most mammals, basically all organs that mammals have are shared with many other vertebrates. So they are incredibly old. But not all mammalian organs are that old, and the placenta is a very good exception to that. So the mammalian placenta originated about 160 million years ago. So what you have here is a baby echidna hatching from an egg, and it's there as a reminder that not all mammals have a placenta, and in Australia, we still have a lot of mammals that are egg-laying. But about 160 million years ago, indeed, the placenta emerged. And since the mammalian placenta emerged, it also evolved into this incredible diversity of forms and very differences in physiology. But amazingly, the placenta has actually evolved more than 100 times across vertebrates. So all the species that you see here are species that have a placenta. And so sometimes some of you in the audience might be thinking, okay, now I'm not sure I know what a placenta is. So I'm going to define it for you. So the placenta is an organ, and it's complex because it's made by either the fusion or the apposition of both maternal and fetal tissues to allow for the physiological exchange of nutrient, of gas, of water, of hormones. Okay, so it is complex by definition because we do have these two tissues having to come together. And all of these species evolve the placenta, and this is how they reproduce. So in my lab, we focus on two groups, mammals, as you'll see, but also an incredible family of fishes where placentas evolve independently several times. So we decided to use mammals to ask the question, how do we create new types of cells? And then use this very special family of fish, how do we actually create a complex organ from scratch? All right, so now I have to introduce you work that is being led in my lab by Martin and Clara. And this is when we decided to go hunting for cell types in the mammalian placenta. And it was really a little bit of a hunt, okay? Because the truth is that mammals are basically made Mammals are made of the same organs and they're made of the same cell types. And this is actually wonderful for biomedical research, but less so for our purposes. But I thought the placenta might be an exception, and this is why. So I'm showing you here these, the schemes of the placentas in human and mouse that, of course, are the placentas we understand the best. And you have to trust me when I say that these are two of the most similar placentas that you can find across mammals. But even though these are two of the most similar placentas, as you can see already by their structures, they are fairly distinct. And their maternal-fetal interface is already quite different. But now I know there's a lot of data here in this slide. I just wanted to focus on these. So what I'm highlighting here are the building blocks of the placenta. We call them the trophoblasts. These are fetal cells. And this is what led me to think the placenta was a good place, is that when you read the human literature, you see there are three main types of trophoblasts that make up the human placenta. But then when you read the mouse literature, we see the double, okay? So I thought, okay, 3 versus 6, this might be the right place to look for differences. Of course, that the easiest scenario or the simplest scenario would be that we would have cell type loss in the primates, could be. But I thought there's also going to be a good chance that we have some examples of cell type innovation. And this was the best place that I could think of to look for new cell types in mammals. So we said, all right, let's look at the human and the mouse placenta and understand their cell types better. Now, we decided to also look at the marmosets because we wanted to distinguish what is human-specific from what is shared across primates. And because we have seen that incredible diversity of cells in mice, we said, let's look at other rodents, let's look at rat and guinea pig, and let's look at the rabbit as well. And these species, which we call the gliers, are the sister group to primates, okay? So we know already a lot about the cells that make up the placentas of these species from histology, but from other sources. We know about the morphology of cells, their location. We have a lot of clues on their function. So what is it that we are adding to this? We're adding molecular profiles. We're adding gene expression. And this is meaningful because we're going to use this data to reconstruct the evolutionary history of cells. And hopefully, if they exist, then identify instances of new cell types. So this is the plan. And how do we do this in practice? Well, we again go back to single cell data sets. So what you have here is our atlases for the mouse. There's many different types of cells. We have cells from the mother and cells from the fetus, because of course the placenta is a collaboration between mother and embryo. And we did this for all the species that you have here. And so what did we find? On the maternal side, we found the same cell types across all of these species, okay? The same building blocks. We actually found very intriguing differences in the proportion of immune cell types. And this is another interest of my lab, which is to study the evolution of immunology in pregnancy. But this, again, would be a subject for another talk. But then when we went to the fetal side, we saw differences in the number of trophoblasts, okay? This is what we expected to find from the literature, okay? So this is what we wanted to find, and indeed we did. So we had everything we needed to then figure out what is their evolutionary history. So how do we identify, how do we study the evolutionary history of cells? There are many different ways of doing it. One way is by building cell type trees. So I'm going to now show you a comparison between human and marmoset. I'm going to color cells from human in black, marmoset in orange. And so just like the tree that you have here is made using DNA sequences that tell us the relationship between the species, we can use gene expression profiles to build cell type trees that look something like this. So how do we interpret them? Well, let's focus here in the bottom on the immune cells. We see to have a one-to-one relationship. So we have macrophages in marmoset, they're close to macrophages in humans, T cells, natural killer cells, and then all the immune cells are together because they share a common developmental origin. But we don't care necessarily about immune cells here. What we care is about trophoblasts, and what we did find. We found a one-to-one relationship when we look at differentiated trophoblasts. So you see more black because in humans we had a higher number of progenitors. But we found cytotrophoblasts in human and marmoset, syncytial trophoblasts. These are just the main cell types of trophoblasts, the same three building blocks, and they were the same in human and marmoset. So now I'm going to simplify these, and I'm going to show you each cell type by a symbol, okay? So the same three building blocks in human and marmoset. And yes, my PhD student would like you to know that this is work in progress, and he reserves the right to change this history before we publish it. Just a disclaimer. Okay, so we have the same building blocks in human and marmoset. We did a similar analysis for mouse and rats, and we found the same building blocks in the two species, but as expected, many more building blocks, the 6 that we had from the literature. So the really informative species here were guinea pig and rabbits. So what do we find there? We found the same building blocks as in human and marmosets. What does this tell us? It tells us that we have a lot of innovation happening in the ancestor here of mouse and rat. Now I'm showing you these symbols, and this really doesn't do any justice to the beautiful biology that is behind this. So let me show you the biology. This is how the maternal-fetal interface looks in human, marmoset, and now we know in rabbit and guinea pig as well. So we have here the maternal blood and here the fetal blood, okay? And between the two of them, we have a very special cell type, which we call a syncytial trophoblast, and it gets its name syncytial because we have many nuclei sharing the same cytoplasm. So we have the syncytial layer, underneath it, we have a layer of precursor cells, other trophoblasts, and then we have the fetal side. So this is what we find in most of these mammals. So what is it that, the ancestor of mouse and rat decided to do? This is how they transform their maternal-fetal interface. So now we have here maternal blood and here the fetal blood. But now they no longer have one syncytial layer. They created 2 syncytial layers that are transcriptomically very distinct. And in addition to that, they created this other new cell type that's called sinusoidal giant cell. It's a polyploid cell type, even if it just has one nucleus. And so their maternal-fetal interface looks fairly different to the other one. And we estimate that this evolved about 27 million years ago. So when we looked at these species, we were already able to identify 2 new instances of the cell type. And you see here this interrogation point. And that's because we have another cell type, it's called the spongiotrophoblast, that we are still trying to determine when it first originated. Our data suggests that it originated here as well, but we are not 100% sure that it didn't evolve a little bit earlier. So this is still work in progress. So what did we decide to do? Well, we decided to continue and go down with the phylogeny. And so I'm going to show you data from my favorite placenta, and that is the placenta of the sheep. Now, sheep have a very different placenta from ours. So whereas the placenta of mice and humans have one point of attachment to the uterus, in the sheep, you have many. These are all of these dots over here. We call them placentomes. These are the maternal-fetal interface in the sheep. And whereas the placenta in mice and in humans is quite invasive, you have cells from the fetus migrating into the maternal uterus. We call this type of placenta very superficial because it's supposed to be epithelium on epithelium. But yeah, not so fast. This is what the sheep can do. So here is that epithelium, the maternal epithelium versus the fetal epithelium, the trophoblasts. But then they can do something spectacular. So they have their trophoblast cells, and then they do a kind of an incomplete mitosis, and they create a binucleated cell, okay? A cell with 2 nuclei. And then this cell with 2 nuclei migrates a little bit to the other side and then fuses with a maternal epithelial cell. So now we create a cell type with 3 nuclei, but this is a chimeric cell type because it's 2 nuclei from the fetus and 1 nucleus from the mother. And then this trinucleated cell can expand and now creates a syncytial plaque. So it's a syncytium again, as I had described to you, but now it's not only made of cells from the fetus, it's now made of cells from the fetus and the mother. And so when we did single cell, even though we are just starting to generate this data, we were able to identify the binucleated cells, the cells that have both fetal and maternal cells. And so just by looking at the sheep, we are able to identify new cell types. These binucleated trophoblasts, we think originated about 50 million years ago in the ancestor of ruminants. And these fetal maternal cells, these chimeric, these cells that share the same cytoplasm, which we think are evolutionarily younger, and they appeared in the ancestor of bovids about 20 million years ago. So one more species and just another evidence for new cell types. And so what we're doing now is going down and generating data for all of these. So for most of the species, we generate developmental data because pregnancy is a very dynamic process. But for elephants and giraffes, and rhinos, this is data that the London Zoo very kindly shares with us, the sample. So this is just data for adults. So what do I want you to take from all of this? First, the placenta is an outlier, okay? You wouldn't find this cell type innovation in any other mammalian organ, at least that I'm aware of, which means that the placenta is a unique model to study the evolution of cell types. And now that we have so many examples and we're still collecting more, we have power to now ask how and why did these new cell types evolve? And to answer this question, transcriptomic data needs to be complemented with data that tells about regulatory regions. So for each cell type, we need to reconstruct the gene regulatory network that specifies that cell type and then try to understand how it evolved in the first place. And so this is where we are with this part of the project. And now very quickly to finish, I want to tell you a little bit about the work of Sophie and Diego. And this is like the most challenging question. They decided to tackle head on, how do you create a whole new organ from scratch? So what do we need to answer this question? We need a good model. And what is a good model? As I told you earlier, is an organ that evolved only recently in evolutionary time so that we can reconstruct the steps. We also, now that we are imagining about our dream scenario model, we would like an organ that evolved independently multiple times, because that would allow us to ask if we see the same kind of principles emerging every time this organ originates. And if this is our dream scenario, how about the possibility of examining transitional forms, right? Since this is such a complex phenotype, we really would like to look at what intermediate steps look like. And this is a tall order to find a model that justifies all of this, but we think we found it. And we found it in this very special family of fish. They're called the quasilids. So if some of you might actually have some of these at home, because these include the guppies, the platies, the mollies, which are very common pets for people to have. And so this is a very large family, more than 300 species, probably a lot more. Now, these species are all live bearers. That means that they give birth to live young, but most of them do it through the process of egg retention. Okay, so this means that the mothers make an egg. They put all the yolk that the embryos need to develop inside the egg prior to fertilization. So the only thing they do differently, say from a chicken, is that they don't lay the egg. They just keep the egg inside the body, and then the little fish is born. And I wouldn't be telling you about this family if this is all that this family did, but a few species independently, they evolved the placenta, okay? So in this case, the mothers put very little yolk in the egg, because in this species, we have a new organ made of both maternal tissues, which is here, and also transformations on the fetal side. And through this novel organ, the mother is able to transfer nutrients to the embryos as they develop, usually leading to much bigger fish in the process. So what we are doing in the lab is studying 5 independent inventions of this family. 5 independent inventions of the placenta. And one of the things we realized is that each of these inventions of the placenta came with different forms. So what I just showed you was the example of a villus placenta, where we call it that way because you have these structures on the maternal side, and then we have changes also on the fetal side. But other species have what we call, for the lack of a better word, the smooth placenta. So these are the same structures in the 2 species, but here, the maternal follicle wall, which is the maternal component of the placenta, doesn't have this villi, it's a much more smoother surface. So we knew we have distinct origins and morphologies. And now what we want to figure out is what are the cells that make up these placentas. So in order to do that, we go back to our tool of choice, which is single cell. And so we do it, but we always do it for a species that has a placenta and a closely related species that doesn't have a placenta. And so what I'm showing you here is the data for the 2 species together, okay, already integrated. Now, doing this kind of work in fishes is a lot harder than in mammals because we have very little information about what these cells are. So what we do is a subtraction. So we have cells from the species with the placenta and the species without. And so we ask, are there cells that are specific to one species that we cannot find in the other? So I'm showing you here is the data from the placental species in blue, and the data for the non-placental in yellow, and everything in gray means that we find the cells in both of them. So what do you see? You see a lot of gray, which are shared cells. You see no yellow, which means that there's no cells that are present in the non-placental that we don't find in the placental. And then you have a few clusters that are only found in species that have the placenta. And so we think that these are examples of new cell types that allows for the emergence of the placenta in these species. So in this particular invention of the placenta is made by cell types that were already there before, conserved cell types, but is also made by new cell types. Then we went for invention number 2. And here, to our surprise, all the cell types in the placental cell were already present in species that do not have a placenta. So it's a new structure, but made of existing cell types. Number 3, we found, again, conserved cell types, but also examples of new cell types. And these cell types and these are totally different. They're totally independent inventions. And example number 4, all conserved cell types. And I thought that this species here, this would be the one that for sure would find examples of new cell types, because we have this entire villi structure there. But what we now know is that this is made of cell types that are already present in non-placental cells in a simple single layer, super simple. But in this species, they are greatly amplified and now are organized in a very different manner. And we are still going. So I hope I convinced you that the placenta is a unique model, both for studying instances of cell type, but also organ innovation. That newly evolved cell types and organs are very important because it's really by studying recently evolved novelties that we can more easily reconstruct the molecular, developmental, and evolutionary steps that led to these new structures. And then the hope is that we figured out how the new complex structures arise. Are there general guiding principles or rules that we can ascertain? And then maybe a smaller question, but an important one, how often is the origin of new organs actually associated with the origin of new cell types? And clearly we have examples that you can do it both ways. And so with this, I'm going to finish by thanking the people that have done a lot of the work. So Letty led the work on the sex differences. Clara and Marcin are doing the work in the mammalian placenta. Diego and Sophie are studying the fish placenta. Now, Rocio is very special here because she's a UNAM graduate student that very happily decided to join my group, but she's studying transposable elements. So it's a very interesting story, but a story for another day. And I wouldn't be able to do none of the work that I described to you today without our very extensive network of collaborators and all the facilities at the Francis Crick Institute. So thank you so much again for the invitation. And yes, I'm open to questions. Hi.
Regarding the first part of your talk, we used to think that sex difference comes from the sex determining gene that derives from the gonad difference.
Yeah.
Right? And my question is if there is any relationship between gonad development and the sex difference, because there is no comparison between your data and what is happening in the gonad. There's it's going one-to-one, or is it?
Well, you're right. So we basically have the breaking is the initiation of ovary testing. That sets up what's going to happen later in our development. But what was surprising here is that when you look at somatic organs, so not at the gonad, that they really show very minor differences, if at all, between sexes, at least for the organs that we study. But it is that was set up by the gonad that later on will lead to very different hormonal profiles that basically are responsible for pretty much all the differences we see in adults.
Okay.
So if we're looking at gonads, then yes, there's a lot of differences. For a very large number. But there, we definitely need to study at the single cell level because
for instance, in the case of the kidney, the kidney, I don't know if the difference that you see is related with the developmental origin, because the mesonephros is the same that gives the gonad, and I don't know if there is something.
So when we look at that stage, okay, when we look at that stage, we found very few genes were sexually dimorphic. Okay, but our developmental single cell data set was not that early on. Okay, so we didn't really focus on that precise period. But from what we see in terms of gene expression is very, very similar between sexes.
And in the liver, is the set of genes are related with lipid metabolisms? Because many of the hormones are processed in the liver to produce, different.
Yes, I mean, you're right. Yes, a lot of it has to do with lipid metabolism. Yeah, so the hepatocytes are little factories, and they have enough differences that you have a little factory males and a different factory in females. And we see there's a lot in humans because the capacity to metabolize different compounds often different between males and females. And we think that because the liver is behind a lot of these processing differences. So yes.
My last question, just it would be interesting to see what is the difference when you mutate the SRY or SOX9 genes, and if still the difference remains or changes.
Yes, I mean, so our data suggests that pretty much all the differences we are seeing are under the control of sex hormones, which means that yes, if you manipulate SRY, for example, if you would remove, what really matters is whether you differentiate the gonads into testes or ovary. So any kind of mutation that biases the system towards one way or the other should in principle lead downstream to the differences we found. It would be, however, very interesting to actually look at that, and probably I should. One of my colleagues at the Francis Crick Institute is one of the discoverers of SRY, so it's just really a matter of getting together and get the right models to do it. It's a very good point. I think we should follow it up. Hi.
All these differential expression in the liver and in the kidney, are they actually meaningful in terms of contribution to sexual identity, or are they just like the consequence of hormones and not really meaningful for anything?
So we think that the differences we find are, in this case, most only the control of sex hormones. I think this is true for mammals. I think when we look at birds, we see something very different. So birds have a very strong identity. Each cell has a very strong sex identity. So sex differences in birds are a combination of both the sex of the cell, which is there because there's a very large number of genes that are differentially expressed because of the sex chromosomes, plus the influence of hormones. But in mammals is basically the influence of hormones. I mean, you can see this in nature. So there's this duality between how important cell autonomous processes are. And species like beetles and birds and flies have these very strong cell autonomous components. And we know this because in nature, you can find mutants, examples of individuals that half their body has a phenotype of one sex and half the body has a phenotype of the other sex. So there's this very famous image of a chicken, which is half looks like a male, half like a female. They made the cover of Nature when they figured out this. And for a very long time, naturalists have seen examples of these type of mutants in beetles and flies and many other insects. But in a mammal, you wouldn't see such a thing. So let's imagine one of us is a chimera, half XX, XY, maybe, to a sibling fusing in the uterus, it's possible. But then the hormones would basically dictate how the sex bias expression would be. So it's very interesting, this duality between some species really having this very strong cell autonomous response and some not. I actually would love to study how that impacts the evolution, the subsequent evolution of sex differences. But I don't know very well how to tackle it.
When they do transplants in kidney or from male to female, there's nothing happens.
Well, maybe, sure. I mean, these differences are set on the onset of sexual maturity, right? So when individuals are reaching this point and there is this influx of hormones and it leads to differentiation. I think in the case of transplantation, we have just, kind of a chimera. It would be very interesting actually to see if we start seeing a little bit of change. Yes, exactly. It's just a very hard experiment to do in humans, but we could do it in other mammals. Yes.
Thank you for your lecture. I just 3 questions related to the 3 parts that you present to us. The first one would be, can you see mutations that are related to developmental disorders in the brain that are more common in either sex, especially in humans?
I'm not aware, but I mean, we have very strong differences in the prevalence of certain developmental disorders between the sexes, like autism
Yeah.
and other disorders. But well, if we knew the genetic basis of all of these disorders, then we can go back and look. This is what I think is happening in the brain. So the kind of analysis that I did here, it doesn't have the level of resolution that we need, right? Because when I started by looking at the whole brain, then we're looking at all neurons at the same time. And I think the sex differences will be limited to particular brain circuits. Like we know from nematodes and Drosophilus that when you have sex differences in the brain, they are very, very much specific to a circuit. So what we need to do is to basically ask this question at the single cell level in the brain, hopefully the developing brain in both males and females, and look for very small subsets of cells. They might be, you know.
Yeah, I was thinking about specific mutations that are linked
Yeah.
to the brain. You don't see that, especially, for example, in male versus female, for autism related.
Yeah,
genes.
so what we didn't find were differences between males and females in gene expression. But I think that, as I said, we didn't have the resolution. So we would need to go there, but now with single cell data for the developing brain and ask if there were differences there. And then if we know the genes involved, then specifically look for those genes. But we do need that single cell level resolution to answer that question.
Yeah. In your second part, I was thinking that if you put a fetus of a different species in a placenta, maternal placenta, then probably wouldn't attach. But is that related to the cell types or the incompatibility between the species?
It's a very good question. So, you know, when now people can do these chimeras where you can take the mouse early blastocyst and then put a lot of rat cells, and now we can actually make chimeras of different other mammals. And the thing is that the cells from the placenta have always to be of the same species as the mother, okay? So you can do whatever you want to the cells that make up the embryo proper, but the trophectoderm, the trophoblast has always to match in with the mother. So we know that has to do, we think it has to do with incompatibilities, but nobody has really explored. So we just know that it doesn't work. So indeed, because we have that different maternal fetal interface, I think it might not work for that. But even if you do it in a mouse and a rat, which do have the same cell types, it already doesn't work. Yeah.
And the last question would be, it's very interesting to study new cell types and new organs that are developing, but also it might be interesting to analyze what are the molecular changes that would lead to a loss of an organ, for example, the tail of a non-human primate versus the human.
I think you're absolutely right. And I think sometimes these organs that got lost can teach us a lot about development itself. So yes, a next chapter. I need to expand the team a little bit before.
Thank you.
From Zoom, Dr. Marta Vasquez asks, do you have any explanation of why methylation of H3K36 and HK327 are most evident histone modifications observed when comparing males and females?
I do not know why. No, that's the short and long answer to it. I do not know. No, we need to dig into that literature a little bit more.
Hi, thank you. Very nice, fantastic talk. My question is related to the placenta. I mean, can you guess, I mean, what is the reason in the differences in the complexity of the trophoblast between different species? Maybe it's related to the differences in the immune system activation during pregnancy, or?
Yes, I think part of the answer is that. So I think that, so the placenta is a collaboration between the mother and the fetus, but their interests are not 100% aligned. So there are two main sources of conflict that we think exist. One is the one you just alluded for, is the immune system. The immune system is being co-opted to help build the placenta, but of course the immune system is also taught to, you know, identify the non-self and work that way. So the more invasive placentas require the evolution of immunotolerance mechanisms, which is something we're trying to understand. So the immunological response to pregnancy evolves very quickly across species. But another source we think of a little bit of, people put it as a conflict, is that it has to do with the amount of resources. So while each embryo wants as many resources as it can get, from the mother's perspective, she would like to allocate the resources proportionally to all of her offspring. So their interests is not fully aligned. And then the embryo's genome is only half the mother and then half the father. So people see the placenta as a little bit of a tug of war. Like there's no optimum, okay? So sometimes you go a little bit more towards the embryo side, which some people interpret as the paternal side. Sometimes you go more towards the maternal side, and you keep moving back and forward, back and forth. So every time we have an example of very fast evolution, it's usually these back and forwards, like we see with the immune system reacting to pathogens. Something of that kind is likely happening in the placenta. And in the process of doing that, then the placenta becomes very, very different across species.
Thank you. And I think I have another question. Can you comment about the role of estrogens and testosterone on the sex bias genes?
All I can say is that they play a very important role. I can say that it's very easy to evolve binding sites for each. Their binding sites are relatively simple, and that gaining or losing a binding site can make a gene be under the control of the different hormones or not. But that's pretty much all I can tell you. Yeah.
Thank you.
I was wondering if, do you have any way of like drawing a timeline between the formation of the placenta, for example, and the ratio of new cell types compared to like conserved cell types? Like, do you have a way of knowing if the beginning of the development of the placenta is only formed by conserved cell types and the new cell types are added in the end?
Wow, fantastic question. I do not know the answer to that. So that is actually one of the questions that Sophie, my PhD student, is trying to figure out. She's studying the development of these placentas and really trying to figure out these new cell types. How are they specified? When are they specified? Exactly when they come up during development. So maybe in a year, I'll be able to tell you a little bit more.
I was wondering, it is often said that temperature is another key factor when talking about cell differentiation. So yeah, temperature. So I was wondering, how did your team manage to deal with this stuff? Because I don't know, maybe temperature can stimulate the many hormones, or I've heard of reptiles that change their sex when they're embryos. So what?
Yeah, that's a very good question. Yes. Well, yeah, temperature is one of the few factors that we know that can alter the pace of development, but not in the species that we are studying. Okay. So in the mammals, and we think the fish, okay, here is a little bit more unknown. We don't think that changing the temperature within physiological limits would really lead to changes. I mean, if you go too high or too low, then it will just impact pregnancy itself. All of these species are kept in laboratory conditions and in temperatures that are close to their normal optima. But yes, you should, yeah, it's a very good question, because temperature in many species does change the pace of development. Yeah, that's just not here. Yes.
Hi.
Hi.
I'm very interested in this topic of placenta. So you said that many species develop a placenta independently. So in all these cases, viruses have an important role in this, or is there any other way to develop a placenta?
I think you don't need viruses for that, even though I know you love very much viruses and their roles. So viruses really do play a very important role in the development of the mammalian placenta, because it is a protein that was stolen or co-opted from a virus that different species use to fuse to create their syncytium. And it's something that we don't know how to explain why, when we look at mouse, and when we look at rabbit, guinea pig, and humans, they all have a syncytium, but they all use different proteins. So it's the same kind of protein, but from different viruses. And why is it that they co-opted different proteins throughout the genome? It's a little bit of it's a question we don't know how to answer, because we think that the ancestor was already taking advantage of viruses. So it already was co-opting protein from a virus to create the syncytium. But for whatever reason, species keep doing this again and again and again, and we don't know why. So this is an open question. But so far, we only have examples of this for species that have a syncytium, but this is not limited to mammals, and as an example in lizards as well, at least. And now people keep looking. So then people ask me, how about your fish? Okay. Now, as far as we can tell, there's no syncytium in this fish. But yes, there's a lot of expression coming out of viruses that now are endogenous retroviral elements. So Rocio, your colleague, will be looking at this, but making a link between the thousands of loci in the genome that are transcribed and the potential functional role in the placenta is not straightforward, but we will try. Yeah.
That's an interesting topic. Thank you very much.
Sure.
We have another question in Zoom. Dr. Vasquez asked, well, she first comments that in the literature, there are several papers attributing the origin of the placenta, at least in part, to some long coding RNAs. Can you comment on that? Could they be important to the origin of the new cell types you are observing?
Long non-coding RNAs?
Yes.
Okay. I think so. I think this is something we need to explore. With the data sets that we have, we can study the evolution of long non-coding RNAs, and we have done that in our past work. So I guess now it's just a matter of looking at them and analyzing them. I mean, it's not going to be one thing, right? Making something as complex as a new type of cell or a new organ involves a very large number of genetic changes, and figuring out the order in which they happen and which one's more important is not straightforward, but it is something we'll attempt to do. So I think, yes, there is a role for long non-coding RNAs in it as well.
Thank you for the talk. I'm very interested about the general principles behind the origin of complex structures. And I think it's interesting how the same set of components or cell types can lead to different behaviors depending on how they connect to each other. So I wonder if you have a think about using something like a networks approach to attempt to model this, or how are you approaching this problem?
Oh, very good question. Well, how are we approaching this problem? We're sort of approaching it very empirically, just by sort of describing what it is that we are seeing. You're right, it's like an emergence of a complex phenotype. So this idea of using network-based kind of thinking is likely to be a good tool. So I guess we'll keep that in mind. Right now, we're just right now our work is really basic, is really just describing what it is there. And we're hoping to describe enough cases that we start seeing some similarities and some patterns appearing more often than others. And once we have those, then yeah, maybe we can do some more sophisticated analysis to try to figure out if there are general principles. Yeah. Hello.
Hi. Well, I once said you convinced me these fishes are a great model. And I was wondering if all these fishes all around are developing placentas, though there are some fishes that have not, and that they may evolve them later. And I wonder if it will be useful to try to find the factors that were necessary for the development of the placenta before the development of the placenta, and if we can find them in another group that has not a placenta yet, it will be evolved. It will evolve it, or that kind of question.
Okay, so when you say factors, at what scale are you thinking? Like ecological factors, molecular factors?
Molecular factors or tissues that are in the proper position, temperature, I don't know.
Yeah, I mean, when we do evolutionary work, we can look backwards in time and, but we cannot really look forward, although we can speculate as what it might be, and we can try to use genetic manipulations to see what would happen if we introduced them in a system that didn't have the same features. So we currently don't have genome editing tools for this family of fish. Is the only thing that doesn't make this family perfect. But as soon as we have these tools, I think we might do something a little bit close to what you're saying, which is try to induce genetic changes to try to reconstruct steps that led to the evolution of a placenta. Yeah, so if you can find the general factors, yeah, this is exactly what we're trying to find in species that have the placenta. And then maybe, yes, if we can mimic some of them in other species, maybe we can get to not a placenta, maybe something a little bit further away from what they have right now, let's say.
Yeah. And just a second question, that is the same one. Because if you were up to look or find these factors, what would you be looking for? Just like the proteins of the viruses, or what?
I mean, at this point, so our plan is to identify the cells that make up the placenta and figure out what makes them different from the species that are closely related but don't have the placenta. Okay, so when we have the evolution of a new type of cell, we want to figure out what are the factors that specify that new type of cell, what make that new cell type evolve. When we have a placenta that evolved, but we think is made of the same cell types, we try to figure out what are the genes that are differentially expressed between cells that exist in both species, with and without a placenta, but now have a very different, lead to a very different phenotype in the species that have the placenta. So in a way, it's like cataloging differences between species. And that basically means that we're going to find a very large number of them. And the difficulty will be to try to figure out what are the most determinant ones. Now, we will try to figure out because these genes, of course, exist in networks. And so if we can combine expression data with gene regulation, we can sort of figure out which genes act with which and sort of identify a cascade. In that case, we would prioritize the genes that are on the top of the cascade, say the main transcription factors. Those would be the ones we'll be looking at, what we think sits upstream of the changes we see. And then we would try to modify them in another species and see what happened.
Okay. Hi, very nice talk. This is a curious question because it is now the function of the fusion of the fetus and the maternal cells to form the syncytium. What is the function of this syncytium in the placenta?
Oof. It does so many things. It basically does all the transfer between the mother and the fetus. In many cells, it also produces key hormones. It's more about what doesn't this syncytium do? I mean, so why is it a syncytium in the first place? Why not just a lot of cells just very close together? Some people speculate that it has to do with not allowing for, the placenta also has to protect the fetus from microorganisms from the outside. And so very few microorganisms can actually pass into the placenta, very few. So the placenta works very well. And part of that is because the syncytium really doesn't allow for space for any other cells to move into the placenta. So it's a bit of a physical protective barrier. At the same time, it is optimized for transport of substances. A lot of the substances that go to the fetus require active transport. And so all of that has to happen with the syncytium. Now, why do we have a chimeric syncytium in sheep? Yeah, no idea. How does the syncytium even look like immunologically speaking, since it has nuclei from two organisms? We don't know. But it is true that we see this syncytia over and over again. Although there are exceptions to this, some mammals do not have a syncytium. I just realized that jerboas, which is a kind of rodent, they seem to have lost their syncytium. So now I'm trying to get samples from jerboas to look and figure out what it is that they're doing. So we know a little bit, but we are still far from understanding all the wonderful things the syncytium does.
Okay, thanks.
I think thank you very much for a very nice presentation.
Thank you so much.